This is TWIS. This Week in Science, episode number 926, recorded on Wednesday, May 17th, 2023. How can a failure be a winner in science? Hey, everyone. I'm Dr. Kiki, and tonight on the show, we will fill your heads with heat, holes, and you. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Think back, all the way back to the first science subject that really got your attention. The first time you thought, hey, this is really amazing, or, whoa, this is really weird, or, nah, that can't possibly be right. Whatever it was, whenever it was, hold on to that thought. That moment way back whenever, maybe when you were 7 or 13 or 39, that moment when your curiosity spoke up, when your curiosity was heard, when your curiosity got answered for the first time. That is what we're trying to bring every week to this show. You, the human listening now, thanks for coming. That insatiable, curious, questioning voice in your brain, that's who we heard. That's who we're here for. In fact, that's the entire reason for This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin and Blair and everyone out there. Welcome to another episode of This Week in Science. Back once again to talk about science, right? Well, that's what we do. We like the science, we talk about the science, and hopefully we get to enjoy it with all of you, and you enjoy the conversation as well. Oh, we've got some good stories on tonight's show. I have stories about black holes that aren't, maybe, confusing comets, and magnetizing your brain for science. What do you have, Justin?
I've got Parkinson's disease causation. More doctors prescribing multiple drugs. This time, the right way. How climate change is causing the hottest days to be even hotter. And the future of materials might be mushrooms.
Yeah, we'll put that all in your head. Blair, what is in the animal corner?
I have bee sweaters. I have hammerheads holding their breath. And I have zebra finches on oxytocin.
Oh, meep, meep, meep. I can't wait that. I love them. I love the little zebra finches. And I hope everyone is looking forward to all of these stories and more, because there's going to be a lot in here. As we jump into the show, I want to remind you that, yes, every Wednesday, 8 p.m. Pacific time, we are here talking about science on YouTube, Facebook, and Twitch. We are TWIS Science on Twitch, on Twitter, is kind of still, I don't know, Instagram, and the Universidon. And if you, I don't know, want to find us, find show notes and all those fun things, you can head to our website, twis.org. And you can subscribe to us. We're a podcast. We're on all the podcast platforms. You know, that's the thing. Subscribe, hit the likes. Help us hit the algorithms in the most positive, optimizing, efficient way possible. Share us with your friends. Okay, that's enough of that. Time for science?
Let's do it.
It's time for the science. Yes. Okay, first I want to start out with a little bit of a confusing comet. It's not that confusing, but a couple of weeks back, not a few, I guess it was back in April, I believe. April, I talked about an asteroid that was acting like a comet, where there was lots of off gassing and stuff that was coming off of it, and how that asteroid was actually the cause of a big meteor shower annually, and all this stuff. Okay, asteroid from the asteroid belt. You're like, great, we get this. That makes sense. Did you know that there is a class of comets that exist in the asteroid belt that do not exist in the outer solar system, in the Kuiper belt and the Oort cloud?
Okay.
The asteroid belt. The asteroid belt. That's like near Earth, Mars asteroid belt. There are comets there. And you have a questiony face, Justin.
Well, I mean, I guess I'm really saying, fundamentally, I don't know the difference between a comet and an asteroid. Because to me, an asteroid should be a big rocky thing or gravelly thing. And a comet is a big clumpy, icy thing. But then when I'm thinking about it, I'm like, well, you know, a lot of the asteroid belt, I would assume, had icy cold things in it. So, yeah, maybe I don't have a good definition of the two.
And they seem to now get that definition is a little bit fuzzy. So we have seen. But the one main defining feature of a comet is that it has the halo, or that streaming tail behind it that we can identify. So when you think of a comet streaking through space, not a meteor streaking through our atmosphere, but a comet streaking through space, is that it is a long, streaky,
and that's water,
object.
Right? The
It's
water
just melted
causes
gas,
and this is-
gassy,
Yes, and it
icy
is
stuff.
yes, sublimating ice, water ice. Yes. And for a long time,
Technical
we
term.
weren't really
Icy.
sure about what was going on, but we got to look at, mass spectrometry. We looked at chromatography. We're looking at all this stuff. We're like, oh, comets, this tail is water ice coming off. And so we've gotten a good picture of that. It was just interesting, wow, there's an asteroid that's off gassing. It's not a comet. Oh, wow, it's an asteroid. This is interesting. But now we have this whole class of comets. We think of comets as these very old, early solar system leftovers. They're like the ones in the Kuiper belt, especially the ones in the Oort cloud. Those are leftovers from when the solar system was formed. So they contain all sorts of old stuff. And we have hypotheses that comets are one of the main ways that water might have been brought to Earth in the first place. And so now we have this small population, there's really not very many of them that we've identified yet, that actually hang out in the asteroid belt. And NASA's been looking at them. And this particular comet 238P Reed, is a main asteroid belt comet. And it is pretty. It's just, if you were to put your telescope on it when it's telescope comet season, you would see a little hazy, halo-y, bluish dot. And that bluishness is also has been artificially enhanced to kind of be the water part of what's happening there. But what it has been identified by the James Webb Space Telescope with its NIRCam this last September, taking a picture of it, and was able to identify that this halo and the coma, it is a comet. It's certainly a comet. It's not an asteroid. So it's definitively now a comet in the asteroid belt with lots of water ice that is vaporizing. And an interesting aspect that they've also noticed that there's no carbon dioxide coming off of it as well. So the question now about this curious comet is, what is this this comet doing in the asteroid belt in the first place? Shouldn't it have melted already? Isn't it close to the sun? When did it form? Is it as old as older stuff? If it's like other comets from out in the Oort cloud, they release a lot of carbon dioxide when they come past the sun and they sublimate and they give off all their water ice. Is it because this particular comet has been warmed too many times and the carbon dioxide's already gone? Or is it that it never had carbon dioxide in the first place because it formed in a place close enough into the sun that only water ice could form and not carbon dioxide, not because it has to be colder to freeze carbon dioxide.
Right. Dry ice.
Questions now. But asteroid comet, not an asteroid, but a comet.
Only raises more questions.
Indeed. And I love that, right? We get a picture, we're able to raise more questions. And we're, as a picture, a picture can tell 1,000 words. In this particular case, maybe a picture increases in the number of questions that scientists might have.
Huh.
Yeah. Anyway.
Very cool. Yeah, nobody brought the story, but the James Webb also found some really big galaxies in the early universe.
Yeah.
And people were already talking about, hey, there's going to be there's a little bit of controversy, like, hey, it's challenging the model of the age of the universe and everything because these are bigger than they're supposed to be.
They're more definitively formed, like spiral galaxies. They shouldn't have that kind of structure yet, but they do, right?
Well, they just re-looked at it, and it turns out that those galaxies that they thought were like, oh, too big, and are actually 10 times larger than they thought they were initially.
So they're even bigger.
Yeah, it's like even bigger.
Wow.
So
there
James
go
Webb
those
is
theories.
amazing in that it is both providing us with asteroid comet hybrid mysteries near Earth and the age of the universe tension.
As far back
So
as
this
we can
brings up
go.
kind of a question I have about the process with these photos. So I feel like it's really common when we get these new photos of our universe to get 2 waves of information.
Yeah.
We get the initial release of, here's these cool photos, this is what we think they mean. And then a few weeks later, go, okay, so we have some updates. Is that because they're releasing the photos to the general scientific community and more people get to take a look at them? Or is it because they're telling us too soon?
Well,
It's
technically, I guess they're telling
think.
us too soon because there are layers of analysis that need to take place based on the way it takes pictures. Also, it's a conglomeration of images,
Right.
And if this was, I think it wouldn't have been too soon. I think the assessments would have been very easy if this was a near galaxy that they had, because then the faint and the bright stars aren't competing as intensely in terms of number of stars and that sort of thing.
But I think
when you're talking about a 13 billion light year away object, it becomes more intensive.
I think in all these areas, any data that's released as it's collected, which even includes visual images that have merely been placed together, what it comes with initially is the speculation that what researchers think it might mean. And very often, I don't know, with National Ignition Facility, with the gravitational wave detector, with any big physics experiment, and lots of science, actually, people have to hype up their results before the results have actually come in to kind of keep everybody interested and keep the machine
Keep the
flowing.
Lights on at NASA.
Keep the lights, keep
publicly
the lights
funded.
on.
Yeah.
And very often, they are exciting. These images and the data and this stuff is super exciting. And I think it's neat when the speculation is less, or is, I guess more conservative than what the actual result ends up being, but it's not always that case. And we often see it's not just a few weeks later. Sometimes the papers that accompany these images or the first release of stuff, sometimes months to a year, sometimes more than 1 year before all the analysis is done. So,
yeah, I just think it's very different from some of the other science that we cover on this show because of that. Because there's these public releases of images, and there's this initial speculation. It doesn't work that way with my hammerhead shark study for later. They don't go, hey, we collect all this data about hammerheads. We haven't done any data crunching yet. We haven't written anything. But
here's a picture of a hammerhead.
Yeah. Isn't that cool? We'll get back to you on what it actually means. It's just very different.
It is very different. Yeah. I think that's an interesting way of looking at it. But I wanted to jump forward from Blair's question about this into also how sometimes things can be seen as failures and then return later. And you've got this cool study, Justin.
Is that did I put that one in the
Are you telling me now that you put it back, that you did put it in the rundown in the right place, and I just segged you to the wrong story?
I might have accidentally got my layout here opposite of the rundown, but yeah.
Justin says, "What rundown?"
last week I talked about doctors behaving badly, experimenting by prescription, by giving people multiple drugs without clinical trials. So I thought this week, I saw this story and I said, okay, here's an example of researchers who are doing clinical trials first. So I thought I would bring this. This is patients with a type of cancer known as hepatocellular carcinoma. They have a tumor type that can be surgically removed. It can be removed by heat, and then they wait. They wait because there's a high rate of this cancer returning within the first couple of years. So just because you went and you got the surgery, you got the treatment, and everything's fine now, high chance that you're going to have cancer again in about 2 years. So they're finding that there's a class of drugs that had been seen as a failed revolution in cancer therapy. These are angiogenesis inhibitors. This is, and I remember, this is from the 90s, but I remember these were, the idea is they would shut down all the vasculature growth of a tumor. So that tumor that was trying to expand and get blood and grow would have all these, the access to blood reduced. Well, problem is, that's when they got into the deeper layers of the trials. That's not what happened. Like life finds a way, so do tumors. And in fact, there was even a study that had shown that compared to the normal messy architecture of a tumor's vasculature with these leaky things that it's trying to create to grab blood, the use of this drug actually sort of cleaned things up, straightened out the blood vessels in a tumor, made them less leaky, allowed more blood to make it into the tumor. So while in some cases they might have worked, and in fact they still are prescribed for like some late stage cancers, they're not very curative. They just prolong life because they actually do slow some of the aspects of growth. Okay, so anyway, this whole class of drugs got basically canned after trials didn't show that they were effective. However, now they're coming back. They're getting a boost from a new set of drugs that are checkpoint inhibitors. These unleash immune system T cells to attack tumors. So what it basically ends up happening is, because the angiogenesis inhibitors kind of clean up the vasculature around the tumors and allow more blood into the tumor, they allow more oxygen into the tumor.
So
which means that the T cells, when they get into the tumor, survive better, are not themselves inhibited by the horrible conditions within the tumor. Also, it turns out by making things less leaky around a tumor, there's less metastasis. Anyhow, they're discovering that the combination of these two drugs is working really well. So, there are currently about 200 drug trials underway to test various combinations of the types of these checkpoint inhibitors and the angiogenesis inhibitors in combination. Guess what? Some of them aren't working. Some of them are having toxic results. And some of them are having really nice effects. The point is, the researchers are testing this in trials ahead of just prescribing it to patients to see what all happens. Which is a good thing. That's how we want science to work.
it's a great thing. I mean, that's what you were definitely asking for last week. So
Yeah.
and I mean, that's what the way that we hope that the science will work, that we'll go step by step to make sure it's safe for people. And that we don't give people drugs that don't work, or give people drugs that work just a little bit, but have all sorts of terrible side effects that make it not worth it. Right.
Yeah. And there's versions of the drug, of each of these drugs that are having different levels of efficacy. My favorite part of the story, one of my favorite parts actually, was that 25 years ago, a Harvard researcher is the one that kind of discovered that this drug that everybody was hoping was going to, sort of be the silver bullet to fight tumors, was having this effect of cleaning up the blood vessels in the tumors. And it was a very unpopular story because, of course, it meant that maybe this wasn't a panacea. And it was a very unpopular story. But actually, if it had been paid more attention to 25 years ago, they might have gotten to this idea of being able to utilize that as a benefit. But because everybody saw that as a negative for what they thought the drug was, how the drug was going to be effective, it went away. So always
it had to go
silver lining.
But it had to go away on its own as a treatment. And then people had to go back to the drawing board until it became that, oh, but what if we put it with this? We already have this thing that does this. And now we could. And so, it's the combination of stuff. And I think we're seeing a lot more of those interesting combinations happening. Drugs being used together that, like, for antibiotic purposes, where one old antibiotic doesn't work anymore, but when you add it to this new thing, it suddenly works even better. Or, there's all sorts of combinations that are happening. And then, like you said, the different variants. So now we have, like, the molecular analysis. So we've got the amazing computer machine learning programs that can go through and go, this molecule with this slight difference right here might be just a little bit better. It's all cool things. But now, Blair, you want to tell us about, I really love the idea of bee pollen sweaters. Little sweaters for bees.
Yes.
But, I have a feeling this is not a good news story.
Well, normally it's fine.
It's not like sweaters for penguins. So.
Right. This is a North Carolina State University study. They found that carrying pollen, as bees are wont to do, increases the body temperature of the bumblebees. The pollen packets that they carry can weigh up to 1/3 of their body weight while they're carrying them. And after they accounted for environmental temperature and body size, the body temperature of bumblebees were found to go up when they were carrying pollen. They rose about 0.07 degrees Celsius for every milligram of pollen they carried. So fully laden bees were 2 degrees Celsius warmer than unladen bees. That's a pretty big difference.
Yeah.
Again, normally that's fine. But there's this thing called climate change that's happening.
Darn it.
And so it's just it's a concern. It's something that has to be considered with our pollinators, because obviously we're worried about pollinators anyway with climate change. This is a whole extra kind of level to look at. Bees are usually very good at cold tolerance. They shiver to warm up when it's cold, but there's very little research on how they tolerate heat. So this could be an interesting impact on bee populations and or pollinator activity. Because, of course, if you think about it a little bit further down the road, okay, if bees that carry as much pollen as they normally do overheat and don't survive, then those traits will not be passed along. What will be passed along will be bees who get less pollen. So it will be evolutionarily beneficial to be less productive as a pollinator.
For the bee, but then we all die.
Correct.
Because we rely on bees to pollinate all of our crops.
Or the plants decide to produce more pollen.
Oh,
so
no.
The probability of catching a pollinator. You think
That's
your allergies used to be bad?
Oh, man. Yes. So the question is, Will they carry smaller loads of pollen? Will they forage for shorter times? Will it overall result in less pollen coming to the colony, with less plants being pollinated in general? Will bee populations decline? These are all questions now that come from this study, recognizing that the pollen sweater does impact their body heat. So there you go.
Will you destroy my sweater? No, yes, this...
Only they could hold that thread as they walk away.
As they walk away, exactly. Poor bees. I mean, it's good for the bees, it's good for the plants, it's bad for the bees. Can't we just stop making the planet so hot? But
that would be great. That would solve a lot of problems that we talk about on this show.
But Justin wants to talk about that later, so we're gonna just tease that Justin has something to say about our warming planet coming up. And
I'm sure it's good.
I'm sure it's great. We can, bring the bees back in, circle it back as we get in there. Okay, I would like to talk not about bees, not about climate change. I want to talk about black holes in space and theoretical physicists and the ideas that they come to when they try to imagine the universe being constructed in a variety of ways. And, Einstein's theory of general relativity predicts that there are giant collapsed stars that become black holes, that there are singularities in space-time that are can be infinitely massive, hold infinite mass, or they're infinitely dense, as I guess it should say more clearly. But we haven't been able to figure out how infinite density can happen, and we haven't been able to actually figure out how black holes all really work. And so researchers okay, this is another part of Einstein's theory being incomplete. We need more information. And so, of course, we need a better idea of how gravity works, because that would tell us everything. And so researchers have been coming up with all sorts of it. There's the Mond theory. There's this other, we've got, just the standard theory of and string theory. So this particular story, researchers have been considering the concept of string theory, which would make space-time made up of infinite amounts of vibrating strings. And these vibrating strings can pop into and out of existence, and then they get all wrapped up together into weird topological conformations that your brain would explode even trying to figure out how they worked. They're microscopically small and can be infinitely complex. And so
technically, there isn't a microscope that could see it.
Thank you very much for clarifying it. And if you've ever tried to untangle necklaces after having them packed in a bag for a while, and it's even worse than that, and it's even smaller. You need the tiniest pair of tweezers to be able to get those things apart. Anyway,
just stop trying to imagine it. You're gonna hurt yourself.
Just stop imagining. And let me just tell you about this story they published in Physical Review D, in which they identify a different option for black holes. The black holes aren't really black holes, that they're a defect in space-time. And this defect in space-time is called a topological soliton. They are, what they say, stable.
Glitch in the matrix.
A glitch in the matrix. Stable defects. So they're just, they're not going to get any bigger, not going to get any smaller. They don't require any other matter or forces. They are fundamental to the natural fabric of space-time. It's like, whatever. Topological solitons. And they should be all over the place. And researchers have determined, though, that because of the nature of these topological solitons in their theoretical string universe, that they would bend light and they would act exactly the same way that black holes do, except that they would not have an event horizon because they're not actually a singularity. They're a bunch of knotted up strings of infinite density. So,
but then they wouldn't have attractive or repulsive forces either, right?
well, this is the question of the gravity and the mass, and they are
Yeah.
infinitely dense, and so it just wouldn't have an event horizon. But so the interesting idea of this is that you could look at a black hole that you think is a black hole, and maybe one day, if we have the right tools to test it, we could actually find out whether event horizons do exist. And, maybe someday we could just like walk right up and just hold a topological soliton in our hand and go, oh, look, it's infinitely dense and I didn't get destroyed. Probably not. But yes, the idea is that so far, string theory has not gotten a lot of support from evidentiary
it's kind of difficult to
experimentation.
prove and all.
So far, standard model seems to be working really great. General relativities, we've got a lot of support. We've got there's a lot of support for like MOND, I think, but we're still looking for stuff. However, the ideas of these topological solitons and other what could be considered very exotic objects could give opportunities to scientists who may potentially want to compare the ideas and test them. So maybe there will one day be experiments that we can actually do to understand the universe better and how these ideas of infinite density may be rectified at some point.
What if they're both right? What if some of them are black holes and some of them are topological solitons?
And that could be true. Maybe it's a both and. Yes.
It's just everybody's right until we can prove it otherwise. You know what? You're all right. It's fine.
Yep. We need until we
theoretical.
It's all theoretical at this point, until we can actually, test string theory. This is still. But I love the idea of, black holes, glitch in the matrix. It's a topological soliton. It's just a bundle of infinitely dense, knotted up strings.
Right.
It's fine. It's fine. It's nature, everyone, or not. We haven't tested it.
I'm actually concerned with the concept of not being able to test
at this point in time. Not yet.
Well,
it's theory. It's
theory. Yeah. So,
the math works.
Sure. Here's the thing. The math works in a model. Fine. What you then do is you find all the ramifications of that math elsewhere. So you don't have to stare into the abyss of a black hole. You can look at the motion of a distant galaxy, right? To see the gravitational effects or how would this have to affect its neighbors? And by looking at that throughout everything, the idea that black holes don't grow or don't accrete or stay the same I don't know. That didn't make any kind of sense to a simple farm boy like me.
But
what if the strings are dipping in and out of our universe?
What do you mean dipping in and out? Stop it. No, you can't keep making up new rules. That's why I don't like playing checkers with you. You keep all these like, oh, now this piece didn't exist. You can't jump it. Oh, wait a second.
And in the
you
end,
put it back on the board, and I see what you did there.
Right, and in the end, this all comes down to at one point Feynman got mad at a interviewer who wanted him to explain something, and he's like, "No, look, I can't explain it to you. I don't understand it really myself, but to explain what I do understand, I'd have to teach you all of everything that anybody knows for you to be able to understand it." And that's the level that we are discussing this at this moment in time. There is a lot of math and there's a lot of theory that is underlying this entire concept that there are experts who really, really do understand this, who can answer these questions much better.
Right.
Maybe. And even if we got them on the show to talk about it, they still could not explain it to it.
Well,
to
and
exactly.
I think our brains would explode by about minute 5 anyway.
I don't know. So at some point though, you would have to get to a point where, like, well, if this exists, then there's no such thing as gravity waves. Okay, wait a sec. We would have gravity waves.
We still do. And they said because of the way that light passes and the way that space-time is deformed around these topological solitons, the Event Horizon Telescope, the gravitational detectors that we have, they can't tell they at this point still can't tell the difference. So there's nothing we have at this point in time that could tell the difference. It's just all in the math.
Right, and that's kind of my point about like they could all be right, is how much of this is actually mutually exclusive in terms of theoretical
and
physics avenues.
I would guess a lot of it, because they all have fun talking about black holes, and they can't all be right about that.
But some could be some things, and
We know
others
they're
could be
there.
other things.
We know the black holes are there. So
No.
are all the black holes the same? Right, like to your point, Blair, are some of them different?
Well, and could the standard model exist and string theory also be true?
Okay, so here's what we do. We take all the physicists with different ideas, we give them each their own universe.
Yeah. This is kind of my point. At this point, it doesn't matter. You're all
I think we could, instead of their own universe, we just give them their own magic 8-ball.
Yeah.
I think you need a prediction, and I think you need to be able to verify it in any form of science.
Absolutely. Well, yes, they're verifying it theoretically.
No, you can't. No, stop it. You can't create your own universe to verify your hypothesis. That's not fair. That's
Tell me.
And with
You
one
need to
fell
be
swoop,
experimental.
Justin takes a hit to all theoretical physics.
We've actually had this conversation before, I think.
Yeah, I think we have.
You want to talk about something real world, Justin? Tell me something.
Oh, what have I got? What did I talk? Okay, so this is another mystery. Currently, Parkinson's disease. Very much a mystery disease. We kind of know how it manifests, but overall, our knowledge is not great. And it is also likely the fastest growing neurodegenerative disease in the world, having doubled in global prevalence over the past 25 years. It's also not great. Although it's kind of interesting because in some reports it seems that the new accumulation rates are slightly lower than so, and the population of the planet doubles about every 25 to 50 years. I don't know. Anyway, there's been some indications that have found some early onset affecting neuronal tissue of the gut ahead of migration into the spine. There's some forms that are associated with rapid eye movement sleep disorders. Actually, majority of them are, which may be a precursor. Some others don't have that as a problem. The root cause, though, is unclear. 90% of Parkinson's cases cannot be explained at all by genetics. They don't have markers or risk factors that you would have for cancer or for some other drug or for some other disease. There's about 10% where they found a link, but then if there's only 10% that seem to be linked genetically, it makes you question whether that 10% is actually linked genetically. Even then, you start to go, well, wait, how can you have a disease that is... So they could be susceptibilities, they could be that sort of thing. But if it's only 10% of the cases, then it's not a clear signal. It's not a good place to attack. Okay. So that pushes it into the direction of an environmental cause. And there have been associations with microbiome that could be associated. People have tried to associate diet to Parkinson's. There have been a lot of attempts to associate different things. An epidemiological study published in... Epidemiological, thank you. Study published in JAMA Neurology by researchers at the University of California at San Francisco has found at least one environmental signal that seems exceedingly strong. Exposure to the chemical solvent trichloroethylene, which apparently is common in soil and groundwater, so what they did is they found this. There have been some lab tests in the past on animals that show that this trichloroethylene can be associated with symptoms that are like Parkinson's. They found that there's a military base, a Marine Corps base, Lahoon in North Carolina, that had a massive problem with trichloroethylene in the drinking water, to the point where they've shut down the wells at the base. They stopped using the water. But for about a decade, at least of those that they're studying here, they have 10 years of people who trained at this base, bathed in this water, drank this water, were exposed to trichloroethylene. They have a 70% higher risk of developing Parkinson's disease compared with similar veterans who trained elsewhere. Camp Lejeune contingent had higher rates of the symptoms such as erectile dysfunction, loss of smell that are early harbingers of Parkinson's. So
I just Googled trichloroethylene out of curiosity, and it says that it's already known to cause kidney cancer, and is also associated with eye and respiratory tract irritation, cardiac dysrhythmia, and has been found in typewriter correction fluid. So that's part of your reduction in Parkinson's in later years. Paint, spot removers, carpet cleaning fluids, metal cleaners, and varnishes. So if it's already known to be cancer causing, that also probably means it's being used less. I would hope. So that would indicate these kind of numbers that you're talking about of the Parkinson's cases being less over time.
Yeah, so exactly. So also it's heavily used in degreasers. It was used in something that was used on children. No, I don't have it here. Yeah, this is a study. This is a great study of people who they know were exposed. Health records of nearly 85,000 Marine Corps and Navy personnel who were stationed for at least 3 months at the camp these many decades ago all show this 70% higher rate. So, but yeah, it's one of the tricky things too, because there's always attempts to correlate Parkinson's to other things, whether it be to nutrition or to diet or to relationships to other diseases. The problem is if it's an environmental cause and these other diseases are perhaps have a genetic basis or a genetic susceptibility risk, and Parkinson's is more directly an environmental trigger, then you're going to have increasingly difficult comparisons of, it's correlated and, it's strongly correlated in this study over here in this area where there's a lot of it in the soil, and it's not correlated at all in this other study where there's tons of, right? And so then you start to have, this is what, when you look around at Parkinson's correlative studies, they can be vastly different when you're trying to compare it to other diseases or diets or other things. So, knowing that it can definitely be an environmental factor, untethered from genetics, is an important
for sure.
thing to know.
I think that's one of the biggest parts. And as far as I know, I think our government is looking. It's already been noted as cancer causing and something to use less in products. That products shouldn't have it in it. I think it's been. Is starting to be reduced as far as I am aware. Tell me if I'm wrong here. But
it says it's banned in the European Union.
Yeah. Okay.
And has been banned in Minnesota and New York in the U.S., but not by the federal government. Not
federal government. Okay. But it is, here, up to 1/3 of U.S. drinking water is, it's detectable in, but not just the fact that it, like so many, like, PCBs and so many other persistent molecules in the environment, the fact that we have lots of jobs where people are using these volatile compounds, degreasers and detergents and various things to get, and they're working in these jobs possibly in the heat and without the proper respiratory protection. And if this is something that you're doing day in, day out, not just
also,
that it's in the ground or in the water, but you're doing it for your job, and that's a job hazard.
Also, it was used previously for making decaffeinated coffee, which I thought so. That's something that's interesting because I remember. I thought, gosh, it couldn't have been Parkinson's. It might have been Parkinson's, or maybe it was Alzheimer's. But there was like this strange correlation between neurodegenerative disease and people who drank decaffeinated coffee, and this might be part of that. Also, I found the thing. There was so probably not a very common exposure, but there was a surgical anesthetic used for children and women during labor that also contained.
Yay for awful, noxious things that could cause us harm. Woo! Come on, people. Let's get it together. Let's clean up our act here. Not with bad things, but with better things that are sustainable.
Not degrease.
Do we really need to degrease that badly?
But the great thing is, when it's an environmental issue, they can be avoided. Steps can be taken to limit exposure. Which, if it was a genetic disease, there would be nothing you could do. Basically, at this point.
I don't know how long ago that study was that you were looking at about decaf coffee, but I'm seeing stuff much more recently that shows that decaffeinated coffee improves brain energy metabolism, may protect brain health.
You know who's putting that out?
Yeah.
You know who's putting that out?
Big
decaf. Well, so actually, there's a couple things. There's also a chemical that they use that apparently to make decaffeinated coffee that's also used in the production of what is it? Methamphetamines, the drug. So these places that were making decaf coffee, were all getting raided and broken into in the early 2000s or whenever, a long time ago was. So now they're using mostly they've converted to high something fluidic CO2 extraction, which is room temperature CO2 superfluidic something or another that's toxic free. They're not using it's not a chemical based separation.
It's just pressure or
Right. So maybe now decaf coffee is fine, but the oldie time method of making decaf
used to be trichloroethylene, and it was bad
a lot
for
of other
you.
nasty chemicals.
Yeah, not so great. Yeah. For 100 years, TCE was used to decaffeinate coffee, but they don't.
And that caffeine that's extracted from the coffee was what was put in soda. So then there's probably trace amounts in that as well.
Wait, what? Yeah. Was it really repurposed? Yeah. I had no idea.
Yes. There's that. Oh, yeah. And then there's the cocoa leaves that Coca-Cola imports to New Jersey to make the cocaine that they don't use in the Coca-Cola. That's a whole other story. Let's talk about how you are everywhere.
I am everywhere. We're everywhere. We're on the internet. We're on the whole world right now.
You're not just detectable on the internet, though.
Oh.
You're detectable in the air. You're detectable in the ground.
Oh, we talk about environmental DNA?
Yes, we
if we can tell which fish are in the ocean, then I would assume you can tell where I'm at.
Exactly, but not just that. They research teams are now talking about what they call human genetic bycatch. So when they go sampling the environment for microbial DNA, for fish DNA, for bat DNA, whatever it happens to be, and people, we're everywhere. We're all over the place, and we are genetic bycatch, mucking up the works for the researchers. However, we could also be adding a lot of information and giving advances to fields like conservation, epidemiology, forensics, and farming. So, for instance, being able to, be able to track ancient DNA and old footprints. I don't know if it wouldn't be preserved that long, but it's just a fascinating idea. But researchers have just published their work on human eDNA, and they have collected samples from oceans, rivers, urban, rural, sandy beaches. They did the air as well. They went to a remote mountaintop along a river, winding along a remote mountaintop in Ireland. Rural villages out to sea. There were towns of thousands of inhabitants. And the only place they didn't find the human DNA was at the very beginning of the tributary where that river started. That was the only spot that they could not actually find human DNA. But the interesting part is, from some of this DNA, yes, people can be identified if we could put enough genetic sequencing together to be able to get big enough and identifying types of pieces of DNA. We could be able to identify people, individuals even, which is an ethical aspect to how this work could impact things. But
you see somebody at a crime scene just pulling a plastic baggie through the air, going,
right? But it's not so easy. Yeah, exactly. Easy as all that.
I think this is definitely going to overrepresent people with allergies.
Sneezing everywhere.
People with dry skin also.
People with dry skin. People with allergies.
Yeah.
Maybe just people who talk too much. Maybe that's also who's.
Yeah. What did we learn during COVID? Laughing, singing, projecting. Big spitters.
Yes. But it's just very fun. We're shedding, we're coughing, we're flushing our DNA all over the place, and we are definitely having an impact on the environment, not just with our activities, but also just with existing. And we are part of the that collection of genetic information that is out there in the world. And researchers are going to have to not only just get used to it being there, but also learn how to take advantage of it. But yeah. Anyway, I think it's very neat to be able to sequence DNA. They were able to sequence DNA from people that volunteers left in the sand. And it even yielded part of their sex chromosomes, according to the researchers.
See, originally when I was listening to this story, I was thinking like, okay, fine. It's time for me to just get my 23andMe done and figure out what's going on, because it's everywhere. So I can't stop it. But now, hearing the complete story, I'm actually thinking the opposite, that it's better to not give the world my DNA on file, because we are heading into a strange direction where somebody could figure out where I was.
If they really wanted to, if the stalker wanted to track you that badly and was into that kind of genetic analysis, yes.
Yeah.
But I mean, you don't even, it's like Facebook figuring out who you are, even though you're not on Facebook.
Right.
They could, I mean, that's how they're finding all sorts of criminals.
Yeah,
now.
your digital footprints.
You're right. Yeah. My digital footprint is out of control.
Ridiculous. Also, I would just say, are you planning on being some kind of criminal? Where you're trying to get away with DNA crimes?
No. I just also know that these things are imperfect, and I don't want to show up for someone else's crimes. That's actually what I'm afraid of.
Here's what you do. If you're going to commit a crime and want to get away with it with your DNA, here's part of what you do. You go to the dumpster next to one of those hair cutting places. You know, the more popular, the better. And you just grab a bunch of the swept up hair samples, and you do your crime scene, and then you take that and you sprinkle it all over.
Gross.
Now they have a bunch of unrelated people whose hair follicles and DNA are at the scene of the crime. They got so many suspects now that by the time they get to you, you'll already be old. But then you'll have to spend the rest of your life in jail.
That's my point. If the criminal goes to the salon and grabs my hair trimmings and does that, but my DNA is on file, then I get identified. This is exactly what I am afraid of.
That really is. Yeah.
Thanks for
Anyway.
giving the criminals all the ideas, Blair.
No, it was Justin. Justin did it.
No, this is your idea.
No.
I was just trying to create the scenario in which you went to the hair salon and ended up implicated in the crime because of the criminal using that technique. Look.
This all does, generate the eDNA question. Yes, we're finding human DNA all over the place. Who should be using this information? Who should get these genetic databases? Is this public information? How's it going to be used? What's happening? I mean, the biggest question, I think, for most researchers is going to be how to get rid of the human contamination so they can just make sure they have the signal that they're looking for. And
I'm
I think
looking for dragonflies, not humans.
Yeah, right. Or make sure that because if you're the humans doing the work, how you have to be able to take out your DNA, make sure you're not contaminating it, but what other people are, all sorts of things that are involved. But big thing that people should think about is if you ever bought a jar of air that was advertised as containing the breath of Marilyn Monroe, or the exhalations of Keanu Reeves, or whatever it is, that this has been a product for sale since
no, I have never heard of
anything
oh,
like this.
Come on. This is since Pet Rock days. People are like,
no,
jarring up air with celebrity. This contains air that was, that some celebrity breathed in it. Now we can.
I have never heard of this.
The closest I've ever heard of that is there was a fart in a jar on Last Man on Earth. And
well, I bet you can find any of it on Amazon.
Oh man, that is wild. So you're saying you could test it? You could see if it was real.
Is there human DNA? Is there any human DNA in the air in your jar? And then maybe if you have a sample of the celebrity, you could figure it out. This is a whole new business model. Testing those jars of air.
If you bought a jar of air, you don't want to know.
I don't want to know.
You're buying unicorns and rainbows at that point. You're not.
I'm buying belief. I'm buying it.
Yeah. Oh boy.
And we hope that you're buying what we've got, what we're bringing you here on This Week in Science. Thank you for joining us for this episode. We are partway through the show and have a bit left to go. And as we head forward, I would like to remind you that we are listener supported. And if you are interested in helping us continue to bring you our jars of science, then it would be great if you headed over to TWIS.org and clicked on the Patreon link and decided to support us through our Patreon community. $10 and more, we will thank you by name at the end of the show. $15 and more, and we will send you stickers. You get some stickers every couple of months, which is kind of nice. And we really can't do this without you. And if you can't support us through the financial helping, sharing us with others would really make a big difference. Make sure you give us the shares on the YouTubes and the Facebooks and the Twitches and all the places, the hearts and the likes, and that in itself is a great big help forward. All right. This is This Week in Science, and it is now time for that wonderful part of the show that we love to call Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I'm going to start with a zebra finch story.
Meep, 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 meep.
Meep, meep. Kiki, how do zebra finches learn their meep, meep?
Doodle-doot, doodle-doot. The zebra finches. I think the meep, meep comes naturally.
Yes, you're right about that.
The doodle-doot, doodle-doot, little bird song, that is learned. They have to learn it from a tutor.
Yes, you're exactly right. And so you passed your zebra finch test, Kiki. So this study is all about how young male zebra finches learn to sing via listening to an adult male tutor. It's an individual that they choose to pay close attention to. Usually that's their biological father, or it could be a foster father who nurtures them. And so in a laboratory model, they wanted to see what the neural underpinnings were for social vocal learning. Overall, they find that oxytocin is what it's about. It's the love hormone. It's the attachment hormone. There's a million kind of nicknames for it, but it's basically a social hormone, I would say. And so it essentially tells them to pay attention, or when you remove it from the system, it prevents them from paying attention. And so their hope was, because the way vocal learning works in zebra finches has been compared to many different animal species, including humans, that they could look at the neurochemistry of the social bonds happening with these zebra finches, particularly during language learning, and potentially apply some of that learning to studies on autism.
Oh, whoa.
So, yes,
Okay.
very interesting leap. So let's get into the study itself a little bit. So what they did is they had zebra finches in the lab. Now, up until around day 50, when they're babies, they are making little cheeps and warbles, what they call them subsong, and it's very similar to babies babbling. It's not real words, right? Just kind of similar to the language that they're trying to get to. And during that phase, the male zebra finch will pay closest attention to the song of their father or foster father, basically the male that they are closest in connection to. They can hear other males nearby, but they don't pay as much attention to them. So in this laboratory experiment, what they did is they impacted oxytocin. And so on day 4, they removed these fathers from youngsters. So they were raised only by their mothers. The cages were enclosed in chambers that prevented them from hearing other birds. Beginning at day 27, they were exposed to a series of tutoring sessions by different male tutors they had never heard before. The tutor's cage was placed next to the cage of the young bird, and when it was exposed to one of them, they were given a substance that blocked oxytocin, and then when the bird was exposed to the other, they received a control substance that allowed oxytocin to function normally. And after completing a series of sessions, the pupils were presented with the 2 different levers to decide who to listen to, and so they picked the one where they had normally functioning oxytocin. So that showed that early in development, the juveniles were actually picking who to listen to. They also found
I like you better.
Yeah, they also found that in previous research that whoever they had a stronger preference for, if they had a stronger preference for their father, their song would sound more like their father's when they were adults.
Mm-hmm. Makes sense.
Yeah. So basically, if they mimicked the father more, they became that song became more intense by the time they were about day 100 when they were adults. So yeah, go ahead.
So the hypothesis would be that with the fathers, you'd have more oxytocin because that's a caregiver, and so that would probably influence the offspring learning from the father more than other individuals.
Yes, exactly. Right. And so by taking the biological father out of the equation and putting a foster father in, you confirm that it is a social attachment. It is not a genetic attachment, which is very cool. And then the next stage of this was to actually physically augment their oxytocin receptors. And so from there, they found that the oxytocin was this specific mechanism that was cueing them in to pay attention. And so overall, their suggestion is that the oxytocin system is involved in how animals decide where to focus attention when they are young. And therefore, this is an area potentially of study for the partnering autism research facility in this study, which I had identified and looked up and lost. But it is this Emory University was partnered with a university and specifically a research lab focused on autism. And so they will be working now on exploring these avenues of oxytocin in social learning in children with autism. So that is something that they can kind of explore, and that has to do with their ability to learn and mimic and receive social information.
Yeah.
Right. Who they're
So
receiving it from.
That would be a really interesting intervention.
Get some oxytocin therapy for sure. It'd be pretty interesting to see also what the receptors look like in individuals with autism, right? Are they functioning the same?
I think that's a really interesting question. There is another study that's out right now, and it's, I don't believe, related to autistic individuals, but it is related to a disorder in which the pituitary gland in which vasopressin and also oxytocin are produced, that vasopressin is not produced. And so they have all sorts of behavior. The kids that develop have all sorts of developmental issues and other things. And they can supplement vasopressin with a nasal spray or a synthetic version. And it gets rid of a whole bunch of the problems with blood pressure and other metabolic aspects that vasopressin is involved in. But there are other issues related to anxiety and mental health that are not corrected. And so there's a big question there about oxytocin's role in that as well. So it could be really interesting to see how all of these things kind of tie in together.
Yeah,
especially that developmental aspect.
Yes,
because
absolutely.
What you're describing, if this nasal spray thing is being utilized by an adult, then maybe it's the whatever benefits are there are only affecting the current state and not that whole architecture that was built
the
up
architecture
without it.
Exactly.
Right.
Right. And of course, there's also crazy advancements in being able to diagnose children at younger and younger ages who are on the autism spectrum. And so if you can come in with interventions that will help them cope with their development biologically, that can really change things for an individual and help them to function much more effectively as an adult. So yeah.
So that's
all about the tools.
It is then you have this really interesting nature versus nurture question, which is if you can intervene and create architecture.
Yes.
Is the false synthetic nurture, I suppose. Does that create an architecture that overcomes the nature aspect of it?
Right. So so that's exactly it. You don't have to it's wrong to say cure autism, right? Because that's not what's happening. People on the spectrum, just their brain works a little bit differently. So if you can figure out exactly how it functions differently, and they can learn how to adapt to that different functioning brain throughout their childhood, then it sets them up with a whole different set of tools as an adult. So you're not like fixing anything or changing anything. You're just giving them tools to be able to learn social constructs easier as they grow up. Right. Yep. So
very interesting.
Yeah. And my second story for the Animal Corner is definitely one of my favorites I think I've seen this year. So hammerhead sharks, they have something in common with marine mammals. Any idea what that might be?
No.
They live in the water.
True. It's all right. You're never going to guess it. They hold their breath when they dive.
I'm picturing another, like two sharks, like in a Far Side. And one of them's holding his breath when he dives, and the other one's like, hey, you really don't need to do that.
Yes. So it turns out they do.
Oh, Gary Larson needs to. Yes, where is
Yes, but they do need to hold their breath. This is what this study has found out. This is a study published this week in Science by University of Hawaii at Manoa. And they found that scalloped hammerhead sharks hold their breath not because they need to, they get oxygen from the water, they have gills. It's because it's cold down there. They're from Hawaii. The surface waters are very warm. They're very pleasant. But when they dive down where deep sea squids live to catch their dinner, it's very, very cold. Yes, basically, they get brain freeze. And so the gills are this huge membrane that, just like our nasal turbinates, we can heat or cool air through that. So when the water crosses the gills, it's all this crazy surface area. And so that can rapidly cool blood, muscles, and organs. They have to close the gill slits during deep dives in cold water because as deep as they go, they can be as cold as 5 degrees Celsius or about 40 degrees Fahrenheit, which is a lot colder than the water at the surface around Hawaii. And so that is a huge temperature gradient. So in order for them to be able to dive down there and come back up and not lose a bunch of energy, like all the energy that they got by the food that they're diving for to begin with, they need to be able to hold their breath to hold on to their body heat. And so they have a feeding strategy that ultimately is similar to marine mammals because they go on these diving trips. They hold their breath for about 17 minutes. And you can actually see through monitoring that they did and videos that they took that they actually respond to how deep they're going, and then all of a sudden their body temperature dips when they start to get towards the surface because they're like like they finally get to open up their gill slits and start doing some gas and temperature exchange. And so their body temperature dips not when they go down to the deep depths, but when they are coming back up closer to the top. Yes, absolutely.
That's fascinating.
Yes. And so the reason that this is relevant, besides just the fact that it's so cool and, oh my gosh, what other fish hold their breath when they dive? I need to know this immediately.
I'm sure all other ichthyologists need to know this as well.
Yes, this is fascinating. I love it. Anyway, besides just wanting to know that for general biology's sake, the other thing is, if you have these sharks diving and they are holding their breath, they are already at kind of a time-dependent and high-stakes scenario while they're diving. But when they are diving, they are also coming across other issues that we have, like deep-sea mining, large-scale fishing, all sorts of things that we do to disrupt the twilight zone where they are diving. So
and
if
especially hammerheads, they like to hang out around the very mineral rich, the seamounts. They'll go from seamount to seamount very often. So they're going to be definitely coming in contact with a lot of that.
Yeah, absolutely. And so if they are coming in contact with human impacts in the twilight zone, does that have an impact on their conservation? Does that have an impact on how effective they are at finding squids and other mesopelagic fish in the short amount of time they have? They're only at that bottom depth about 4 minutes. So in the 4 minutes they have while they're holding their breath and they're in this ice cold water, is human activity spooking them away from a space that is a good feeding ground? Or is it scaring away or changing the behavior of the fish they're looking for? These are questions now that impact conservation of species like this, because if you know that they're holding their breath, you know that the areas where they dive are exceedingly important to protect.
Yeah, because you're going to hold your breath and go, nope, I'm out of here. I'm not going to hold my breath that long.
Yeah.
you'll be really impacting their ability to survive.
Yeah.
So let's stop mining the bottom of the ocean.
That'd be great. Hey, buds, that'd be great.
Yeah, that'd be nice, right? Wouldn't it be wonderful? Let's protect everything and all live in harmony.
Mm-hmm.
I've decided I'm just going to be Mabel from Gravity Falls forever now.
That's a good one to have. Where's your sweater with a fun graphic on the front of it?
I know. I need a collection.
Your loud earrings. Yeah.
Yes. I have pictures of myself with those sweaters and loud earrings from the 80s.
I love it.
Faux show. All right, Justin. As yes, this animal corner's coming to a close. So, what do you have?
I'm going to speed it up here a little bit.
Yeah.
Get through my last couple of stories so we can keep this tight 90. Research led by the University of Oxford has found that climate change is causing the hottest days in Northwest Europe to warm at double the rate that average summer days are getting hotter. So the maximum temperature on the hottest days, the more extreme heat, is increasing at twice the rate that the maximum average temperature is. Meaning that for the, where you're talking northwestern, England, Wales, northern France, areas like this, current climate models are pretty accurate in predicting the rate of warming for average days. They have massively underestimated what the extreme heat of hottest days of warming are going to be. So, if this is an early indication of what is to come, it's going to be bad.
But Justin, isn't the Gulf Stream going to collapse and then Europe will be thrown into a wintry hellscape anyway? So it's fine. It'll cancel out.
It could be part of the, that could be part of it, right?
But it's going to be hot first and then cold.
Yeah. Fire, then ice.
So we talk about, a lot of people talk about, whoa, you're talking about weather, not climate, because that's global. Nah, we're at the point where we're talking about both at the same time.
Yeah.
We're talking about both at the same time.
Yeah, and I do keep wondering with the La Nina, El Nino trends going back and forth. I mean, even here in the Pacific Northwest, we had some of the most scorching summer temperatures that we've ever hit during La Nina, which is supposed to be the wetter, cooler version. And then and El Nino is the hotter one, which is supposed to be, it's the next couple of years are going to be hotter. And I just could, yes, this is the weather and the climate part as it comes together. Yes.
And the other thing is too, this is an area that probably, I've not spent much time in the north of France or the north or south or east or west of England or any of this, but I assume that they probably don't have a lot of air conditioning. Like I'm in Denmark now.
It's
nobody
like
has an air conditioner.
Yeah.
Almost nobody has an air conditioner because it's never hot enough.
Yes.
And so when you get a heat wave, that's when you hear about a heat wave of 90 degrees killed a bunch of people in Europe. Whereas in California summer, that's not even the hot days. Those are the cool days. Those are the days you go outside.
In parts of California. In some parts of California, no one has air conditioning either. And they are also having trouble. Yes.
Right. Yeah. San Francisco is one of those odd places that doesn't have air conditioners. So when you get these extreme heats in places that really don't have the infrastructure for it, it means you can have refrigeration problems. You can have all sorts of everything problems. Anyway, so that's bad news. And a little bit of good news.
Yay! Bee sweaters.
We're in this part of the show. Researchers from the University of Sydney have developed a biological growing process that uses mushrooms that can take some organic waste and make compostable products at low cost as a sustainable solution alternative to plastics.
I like that.
Growing everyday products using the unique properties of edible mushrooms. They start with a mold. No, not that kind of mold. The kind that you cast things into, like not the fungus, but a form. They have a form that they will create, maybe 3D print it, and then they have a mycomaterial, mushroom material that can be a mixture of mushrooms and, say, sawdust or cardboard remnants. Then they allow this material mix to grow, filling out 3D printed forms. They say it's lightweight, compostable, biodegradable, sturdy enough to be used as a replacement for many plastic applications. The mushroom varieties that they've been experimenting with are the types that you can buy in the grocery store. So they're edible types. And they do a good job binding together this organic waste matter into a network of roots as they grow, creating structures known as mycelium networks.
So we know they've got the mycelium networks. We know that fungi can be antibacterial depending on the fungi that you're talking about. But if you're creating this thing, are they all dried out and that's why they're good as plastic replacements, or are they are they going to grow bacteria? I mean, can you wash it once? I don't know. Is this the beginning? What's this the beginning of?
If it's compostable, I'd imagine you can't get it too wet.
Right.
Yeah.
And I mean, can I use it? Do I make it at home in the old yogurt maker that my mom had from the 70s? And then, every morning I have a new coffee koozie.
So yes.
Is this the way it's going to be? Make it at home.
The answer is yes. To one aspect of this, which is that while they developed this whole thing in a lab using, design software and 3D printers and all this sort of stuff, they actually are provided in this paper, or in the paper or at the website, methods for making it at home.
Okay.
Making the basic mushroom and growing the mushroom and combining it with a blender and some other stove top kind of stuff. You can do this at home. So they're playing around with it because this is right now they've got this material and it's a matter of trying to figure out what to do with it.
Right.
So they created a flower pot they grew it around sensors, electronics. So they created a flower pot that has sensors built in that automatically monitor and report the moisture of the soil content. They say, oh gosh, hey, this has got some pretty good thermal properties. So they made a coffee cup holder because it insulates it. They have a textured pot plant that can decompose as the plant is getting ready to grow into a larger pot. So they're really playing around with this. And of course, they're also working now on getting a version of it that itself can 3D print.
What?
So that they can make materials, 3D print materials with this mycomaterial, mushroom-based material.
Right. It would have to somehow, connect. What kind of glue do you use? What is the stuff that makes the mycorrhizal stuff stick together in the first place? And okay, this is interesting.
So I will say, at first I was kind of wondering what was new about this, because I know mushroom packaging exists. We have it. It's available on the market. I wish it was used more, but I definitely have bought things on the internet and they've shown up in mushroom packaging and I get very excited. But what this sounds to be innovative around is like the use of it for, first of all, things that generally speaking you need to use plastic for. And second of all, things that are incorporated into other technology. It's not just a static thing. And so that seems very exciting. Although
they did say that one of the highest and best uses of it probably will be packaging.
Yeah.
replacing cardboard boxes and stuff. Because
more of it. Yes.
Basically the idea is this is something that is sustainable, renewable, decomposable, unlike plastics. that also doesn't require cutting down trees and this sort of thing.
It's not. You're using the decomposers in the compost. Mushrooms are decomposers, so it's actually not just decomposable. You are adding decomposers.
Yes, you're enriching the compost. Yeah,
you're making it more.
Yeah.
So the
Plus,
level,
fry it up, put it on a burger. No.
The level to which you incorporate it with other materials, like maybe it's the sawdust or the cardboard or whatever it is, can be based on longevity. So you might have an electronic device that is the sort of thing that, there's plastic humidity detectors. You got plunked in all of the plants in the house. Okay, so those could be maybe replaced. The plastic packaging or plastic wraps around packaging to preserve it. There's a bunch of different ways. Anyway, right now they're just playing with the possibilities. But I like the idea that they are creating a version of this or a recipe of this for people to try at home.
Yeah, that's very cool.
Like we have some ideas, but here's how to do it. Go out there and come up with more.
Do more things. Try new ideas. It did look, there's a video that went along with that story that looks as though they're using lost wax casting to be able to create some of these forms, which is pretty fun in itself. Just, being able to use lost wax casting for mushroom stuff. People are going to live off the grid in a very wonderful way someday. Eric Knapp asks, is this bad for people who are allergic to mushrooms? And probably, I would imagine, I don't know. That is probably the thing that has to be tested.
If you're allergic from touching mushrooms, it depends how allergic you are.
Is that a thing?
You can't
I
eat
didn't
them,
even know that was
you're
a
probably
thing.
fine.
I think it's a thing.
I didn't even know that. Is that a thing?
It's a thing. It's a thing. It is a thing. Moving forward, I have a story about magnetizing your brain. So, in the past, we've talked about transcranial magnetic stimulation, and people use it for a number of different things. The primary use that people are interested in it for is in the treatment of depression. And some people use transcranial magnetic stimulation, and they do bits at home. There's actual clinical transcranial magnetic stimulation that is done that has a number of sessions, can be done like daily for 10, 20 minutes. And it seems to be a fairly intensive production to get involved in the treatment of what's going on. And some researchers at Stanford University wanted to look into how this neurostimulation, targeted magnetic neurostimulation, is actually working in the brain, or what is it doing in the brain? And so their paper was just published this last week in the Proceedings of the National Academy of Sciences. And this is an FDA-cleared treatment that they looked at, known as Stanford neuromodulation therapy. And in effect, they were able to combine transcranial magnetic stimulation with an additional, this Stanford neuromodulation therapy that does high dose magnetic pulses, and it changes it so you can do fewer sessions, just 10 sessions a day for 5 days. So you don't have to do it long term. It's shorter term. It also made it easier for them to see if it worked for their study. And anyway, the idea is that normally in the brain, you have sensory information coming in. Sensory information gets processed by the amygdala, and then the amygdala and other areas will push out to the cortical areas, and the cortical area goes, what should we do about this? And then you get the information coming back telling you how to respond, and everything is rational and nice and not overly emotional, and everything's great. However, in the case of depression, what has been kind of seen to occur is that, and especially when they looked at their data using fMRI imaging of the brains as they had people in this treatment, they saw that people had an area, this anterior insula and the amygdala, it was integrating the sensations, and instead of in people without depression, it was going to the cortex, and it was great.
The deciding part.
But the depression, the people with depression, it was as if the cortex, the deciding part, already had its mind made up, already had a mood, and was pushing information back to that insular cortex. So instead of information going from the sensory out to the deciding and then back to the sensory, it was just like the deciding part was like, nope, we're depressed. We don't like things. It's bad. We're pushing it down, pushing it down, pushing it down, pushing it down. And so they showed in their treatment that when they treated people with this transcranial magnetic stimulation with their neuromodulation, they showed that they were able to reverse the currents of neurological activity within the brain. So that people with depression who had this kind of reversed set of neurological signals, that subset of individuals were really, really impacted by the magnetic stimulation, and that their depression started to alleviate, and they actually showed a complete change, a switch in the direction of the energy flow within their brains.
So, okay, so I'm missing one part of this. I get that the amygdala, which is this primitive reactionary sort of...
All of it in there. Yeah, all of it. The insular cortex, the amygdala, all of it. Yes.
So you got this amygdala thing, and it sends out the signal, because the amygdala is also highly implicated in stress and anxiety disorders and post-traumatic stress disorders and this sort of thing, because it sends out like an alert of some sort. Now, they get to the prefrontal cortex. It's supposed to decide whether or not this is useful information, but it doesn't talk back to the amygdala very well. It can't really tell it like, hey, quit sending the signal. I got it.
Right.
The amygdala will just keep sending and sending and sending.
And sometimes it's like, shh.
Is this silencing the amygdala, or is it?
Yeah, it's not. Yes, it's silencing those sensory areas that are. It's like the like you said, the cortex area is sending the signal,
trying to send it back,
trying to send it, and basically it's as if the brain's mood has been decided already, and it's just like, nope, the cortex, the deciding area, is ignoring this other stuff that might potentially make us happy, or this other stuff that might potentially make us want to do something, this other stuff that would potentially be stimulatory to what our life is. Nope, push it down. And but what the take home of this is that it's not something that they're not saying that this neuro modulation through magnetic treatment, that it should be something that everybody should get who has depression. What they're saying is that they actually saw these basically like signals within the brain within a subset of people who were depressed, and that those people who had that kind of reversed activity in the brain were the most likely to have the most positive result from it. Okay. And have their depression alleviated.
Right. There's different causes of depression. And so this is identifying a very specific cause and a very specific treatment.
And a very specific treatment. Right. It doesn't get rid of, my dog just died, or, the life things that happen that go on to cause some aspects of depression. There is also,
or your serotonin receptors are broken. That's not going to fix that.
Right. There are so many things, right? There are internal, physiological, metabolic, psychological, sociological. There are very many aspects. This is one particular subset, and it happens to be that they can see it in an fMRI, and that once they see it, they can go, this treatment could really work for you.
So I think this is a really interesting point about treating depression in the future, and that like this could help get us there. Is that currently it's much more complicated than this, but essentially if a professional says you have depression, they say, let's try this drug. Okay, that didn't work. Let's try this drug. Okay, that didn't work. Let's try that. Right. So it's kind of more of a, based on what you're telling me, we're going to make this best guess on your treatment plan. But if you could actually identify mechanically what's causing your depression, you could have a tailored treatment that is much more likely to act effectively and quickly and without a bunch of weird side effects.
Yeah, and there was a weeks ago, I guess, I had a study that had a microbiome effect.
Impact,
right.
Which, of course, because the gut is lined with
the gut is your brain
cells.
Yeah.
Your gut is part of your brain.
It's part of your brain. Wait, what are you saying? Yes, your whole gut from your esophagus down to when your poop comes out is lined with neurological cells, which are connected to the whole central nervous system and everything else too. So, so we're brains all the way down. So what affects the gut, what's affecting the brain, what's affecting the sensors, what's affecting all of these things. And so it's all a spectrum of order and disorder within each human.
The medical technology is astounding, and it saves many lives, and we have come so far. And I am thankful and grateful for all of the things that medical science has done to this point. However, there's still a lot of symptom-based guesswork.
Guesswork. Mm-hmm. And when you have actual biomarkers that you can look for,
absolutely. Yeah, and that's what like modern science is getting us to, is actual mechanics of what's wrong in your body. And the more you can tailor medicine specifically to a body, the better treatment's going to get. I think it's really exciting.
I think it's very exciting. And speaking of, I don't know if this isn't tailoring anything to anybody's body, but in case you were wondering whether that pandemic brain fog that you maybe thought that you had for a bit in there, what? I can't think of things, and I'm just not as clear. My decision-making ability is not that great. And well, Ohio State University researchers used a small group of students comparing the results in decision-making from their 2020 group of students, the 2020 fall semester group of students these are students in the thick of it, probably doing online college,
for sure. Yeah. Fall 2020, everything was shut down.
It was shut down, right? Yeah. So they compared the decision-making of students participating in their research to the decision-making that students in previous years had, the abilities that they had. And they found that the hypothetical situations that the researchers put in front of the students in 2020, they found that those students were more likely to kind of go back and forth between either just going with their gut and just being like, whatever, they're just going with their gut, or really, really just be like agonizing, mulling over, thinking about the things, depending on how things had been described to them. The researchers say that what the big important finding that they came to is that the stresses of everyday life can wax and wane, and they can potentially overwhelm your cognitive resources. And you can see real downstream effects on everyday activities that require your energy and your effort. The study provides additional information to understand why students may have been having difficulty coming to class, focusing on class, and getting things turned in, because there was this global event affecting every part of their lives. And so, this data that they have come to report from their samples of about 722 undergraduates compared to the 161 who participated during the 2020 fall semesters that pandemic brain is real, was real, and that there is a real reliance in decision making, depending on the energy that you have, whether you're going to be looking at a scenario as like, is this a win or a lose scenario? What is happening in this scenario? And that the researchers are not researchers, but students or people are not necessarily adequately perceiving risk because they're thinking about things either just going from the gut because they don't have the energy to deal with it, or basing it only on their experience. And the researchers said that despite there were some inconsistencies between the 2020 cohort and the earliers, and they said that regardless of those inconsistencies, the students in 2020 were just as confident as pre-pandemic students in their decisions, even though they were making them differently. And I think
I'm still trying to figure out how I ended up with pandemic brain like a decade before the pandemic hit. Like, is there any explanation for?
So this is what I am taking from it that I think is the very important message, is that for the first time, we were really seeing the large societal effects of massive stress on lives and the ability of people to make decisions. Whereas there may only be certain subsets of human populations that constantly exist in those situations, those in poverty, those in poor health situations, or I fully believe that the pandemic brain that people are talking about is we're talking about it. It's just the brain, and it's the brain on stress, and it's oh goodness,
everyone was experiencing the same stress at the same time,
and the people who are talking about suddenly it's online because people in media who write are talking about it, intellectuals are talking about it, teachers and their students on suddenly it is a societal thing that
it's a universal, but in any one moment before or now, you don't know what fight or flight scenario an individual's brain is having going on inside that is unique to them. And so in this in this particular situation, everyone had their own fight or flight with no place to fly to. We were all stuck. We had no actions to take, but we had a constant fight or flight signal going in our brain for months. And so somebody else might feel similarly in a different situation, but you wouldn't know to look at that the same way because it's very unique to them. But this is also the joke about why millennials are so anxious and can't make decisions, is because they've spent their entire adult life moving from crisis to crisis.
It's just one more.
So like
again,
like older millennials came of age around 9/11, and then there was the housing crisis, and then there was a jobs crisis, and then there was it's just been something ever since, every few years.
And that's why Gen Z is like, the third child and is just like, whatever. I just can't even.
Yeah. Well, that sounds like Wait, what are we? I forget what generation I'm supposed to be.
You're Gen X, right?
Gen X.
Okay. So Gen X grew up thinking that the world was going to end from nuclear war anyway.
Right.
And all decided to just go party.
And we were all latchkey kids.
You know what? Hey, we don't know when.
Kool-Aid.
The mushroom clouds are going to be popping up everywhere any day now. Let's go listen to some pop music at the club.
There you go.
Pop music. Pop music. Have we done this? Did we do it?
Also,
did we make
okay,
it?
yeah, I think we could be done.
Yeah, go watch some pop-up video. We're done. Remember that? No. I remember that.
What are you talking about?
It was good
What are you
stuff.
talking about? What are you even talking about?
It was VH1 played it, and it was you would watch music videos, and little bubbles would pop up with facts about the music video and the artist.
I remember. I do, but that is
Why?
That's probably
That's
what
VH1. It's not
Is
MTV.
there a second? How did that?
because she was singing pop music, which is I think what they used for the theme song for Pop-Up Video. That's right. And we were talking about Gen X, and it's just it's all there.
I was going to say that. I was just going to say as well.
That was
Keith
Robin. Was it
the
Robin
end of the show
Scott?
ready to go.
That was like 1979.
There's also an endless number of studies relating the pandemic to other things. The vaccines to other things, the pandemic to other things. It's because we all have a shared time point event that we can agree on. We'll start here and study from here to there. It's going to be so much noise from this. I can't wait until it's like so long ago that 3 or 4 more pandemics have happened and we're done talking about this one.
Who cares anymore? That's such old news.
Yeah.
But next week there will be more news, more new news,
Science.
more new science. Yes. I think we've come to the end of the show. Yes.
Certainly.
We certainly have. And I definitely want to say thank you to everyone who has joined us here today in the chat rooms. Thank you for talking about the science with us and for being here chatting. Really love that. Fada, thank you so much for your help with social media and show notes. Gord, Arnlor, others who help keep the chat rooms happy, clean, fun places to be. Thank you for being there and doing your chatting as well. Identity 4, thank you for recording the show. And of course, Rachel, as always, thank you for editing the show. And I would be totally remiss if I didn't thank our TWIS patrons. Thank you too. Yeah, Craig Potts, Marie Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Levin, George Chorus, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyders, Donathan Styles, aka Don Stylo, Ellie Coffin, Reagan, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon Noodles, Jack Bryan, Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, aka Zima, Ken Hayes, Howard Dan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Kurt Larson, Greg Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you would like to help support the show on Patreon, please head to TWIS.org and click on the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from TWIS.org/live.
Want to listen to us as a podcast, perhaps while you knit a nice bee sweater? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twis.org, where you can also sign up for a newsletter if you are interested in that sort of thing.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into the event horizon of a black hole that may or may not be a black hole, that also is part of a string theory layer cake of onion ring shaped tiny universes that may or may not exist.
And if
Never
you are
gonna get
beyond
one.
the Schwartz radius of the black hole, when you cannot escape the Twitter, you can still talk to us at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week